So let's move on to the second talk, if I can find it. Yeah, from Carlos Buesa, from Oryzon Genomics. I just met Carlos earlier at lunch, and I was very struck by how successful his company has been. So I'm looking forward to this talk, which is called LSD1, an epigenetic therapeutic target to modulate different, notice that, neurodegenerative disorders. Carlos, thank you.
Thank you very much. Okay, thank you very much. Good afternoon to everybody. So from the macro view to the micro view now. Oryzon is just a biotech company here in Spain. So we were founded in 2000, 16 years ago. We are actually one of the first startups of the University of Barcelona. And we are already, today we are a public company and developing our own programs on that discovery. So how this is going on? Sorry.
No, to advance the slide, you use the mouse.
Ah, the mouse, okay.
You use the left to advance, and then to go back to the right one.
Okay, the left to advance.
The menu comes up.
Okay, thank you very much. So we are basically working on epigenetics. As you probably all know, epigenetics is basically the way that the cells regulate the functioning of genomes, and they do so by a number of post-translational modifications on the histones mainly. It's not the only way, but it's one of the main ways. And among the different chemical modifications that histones might suffer, methylation and demethylation is one of them, and this is the field where Oryzon has been specialized in the last years, and we have become one of the global leaders in the area. So I'm going to talk today about epigenetics and neurodegenerative disorders. So neurodegeneration and epigenetics have been linked for many observations in the last years. Among them, it's worth to mention that a number of reports have shown that HDAC inhibitors are able to improve the conditions of different animal models representing or reflecting different human diseases. The reason why these HDAC inhibitors have not been able to progress in human clinics is well known. Is because HDAC inhibitors are not selective enough. They have a profile of safety which is not sufficient for chronic settings, and that's one of the main reasons. So we are working on LSD1. LSD1 is an lysine-specific demethylase, which is an enzyme. Structurally speaking, is a fat-dependent monoamino oxidase, and this enzyme is demethylating the dimethyl and monomethyl form of lysine 4 and lysine 9 in histone 3, and by doing so, is reorganizing or rearranging the scaffold of chromatin and setting on, turning on and off a number of genes. Well, we know today that LSD1 is also a partner, a regular partner, very often found in HDAC1, HDAC2 transcriptional complexes that are governing the expression of important genes in neural and glial cells. We also know that has been described recently that LSD1 in suppressor screens, that LSD1 knockout, LSD1 suppression produce, enhance the removal of misfolded protein with critical roles in different neurodegenerative disorders. So we have a molecule which is basically a dual inhibitor of LSD1 and MAO-B, and which has all the properties or the pharmacological properties that you should ask to a molecule to be promoted, to be moved forward in the clinical development in a CNS human disease. We have is oral, we have a good PK and good ADME properties, has a very nice bioavailability, allowing an oral regime of only once daily. It crosses very well the blood-brain barrier, and we know that this has target engagement in the brain, and that is having also a high therapeutic window. So with this molecule, we have been able to demonstrate in 3 different animal models of 3 different human disorders that we have a proof of concept and the animals improve significantly. Okay, I'm going to show you just a glimpse on 2 of these models, Alzheimer's disease and multiple sclerosis today, and to tell you where we are right now on this molecule. Okay. This is an epigenetic modulator, so we were on purpose not wanting to work with transgenic models in which you have an artificial promoter and the chromatin is by definition artificial. So we were deciding to work with a model which was developed in Japan at the University of Kyoto and by the old breeding techniques. And these animals, some 8 animals, develop an accelerated aging. And in this process of accelerated aging, they start to manifest problems, cognitive impairments, memory troubles, which appears normally at month 4, month 5. As long as it goes, the life of the animal, we are able to find other manifestations which are also present in the human disease. So on those animals, what we have done is basically we have treated the animals on month 5, starting treating the animals daily, orally, with our drug for 2 months or 4 months, and at the end of these periods, we were basically scoring the animal for memory performance, and we were taking different parts of the brain to analyze the biomarkers. So we test the animals by a simple test that you all know, which is a novel object recognition test, which scores the capability of the animal to discriminate between an old and a new object. By doing so, what we see on the black column, this is the normal discrimination index of a normal healthy animal. This is the parental strain. In the white column on the right, what you see is the discrimination index of these sampled animals when they are treated with vehicle. And on the blue columns, you see the animals, the litter mates of the sampled model treated with our compound for 2 months or 4 months. As you can see, we are able to restore the basic memory performance of these animals. So that was, these initial experiments were done at 3.2 milligrams per kilo and 0.96 milligrams per kilo. In subsequent experiments, we were dosing lower, and as you can see, we were seeing still full rescue at 0.32, and at 0.11, we start to see a fading off of the phenotype. Okay. So I was mentioning, I'm not going to explain why we were after a dual LSD1 MAO-B, that was on purpose, but we were interested on dissecting the contribution of both arms on our drug. So for doing so, what we were introducing in subsequent experiments were additional arms in which we were treating the animals either with MAO-B inhibitors only or LSD1 inhibitors only. On the left panel, what you can see on the green column is the result of treating the animals for 2 months with a well-known commercialized MAO-B inhibitor at concentration where we know that there is full brain target engagement, 3 milligrams per kilo. As you can see, we see a small trend, but this is failing statistical power. So MAO-B is not really driving the effect that we see on those animals. Well, the question then is, why do you need a MAO-B activity for? Okay, on the right panel, what you see is on the ochre and on the red column, the effects of treating the animals only with LSD1. As you can see, LSD1 is able to do the job, but in these concentrations, the ochre is having the same relative brain exposure than the light blue and the red and the dark blue. What we see is the LSD1 alone is doing the job okay, but is doing the job less efficiently than in combination with MAO-B. So MAO-B is enhancing the combination of LSD1 plus MAO-B inhibition is enhancing the effect. Okay, so what you see here is a recopilation of different experiments that have been done by us and by somebody, and additional people on this strain in the same lab. In the blue, you see the memory performance of the normal parental strain, the normal healthy animals. On the red, the memory performance over time in months of that size of the females and males of the sampled model. So just to make a summary of all the work that we have done, when we start treating at month 5, where we see already a difference between the parental strain and the sampled animal, we are able to restore the memory performance. So in this model at least, these results are suggestive of a possible disease modifier drug. Okay, when we go to the molecular level, if we go to the hippocampus and we make an analysis of the gene expression, what we saw is that LSD1 inhibition or 2001 inhibition was producing an increase on genes which are associated with improved cognitive function and neurological plasticity, which is what you would be expecting. But very interestingly, what we saw also was a reduction on the inflammatory signal. So we saw the reduction of a number of genes involved in inflammation. Among these genes, particularly one was catching our attention, which was S100A9. Okay, S100A9 is a pro-inflammatory molecule, is well known, and it's particularly interesting because it has been described to be upregulated in Alzheimer patients, but also in patients with postoperative cognitive dysfunction. We were hearing about that this morning, and traumatic brain injury. Okay, so there is a relationship which is being worked out between S100A9 and Alzheimer's disease. In fact, in a number of single and double transgenic mice, where there is a combination of the removal of S100A9 either by lentiviral brain injection or by crossing with a knockout mice of S100A9, what we basically get, or the authors basically describe, is that there is an attenuation on the memory impairment, or an improved memory, if you want to speak, and a reduction on the amyloid plaques. In fact, in the last years, there have been also some papers describing a mechanistic correlation, a mechanistic interaction between S100A9 and the beta deposition. Apparently, the S100A9 monomers, dimers, and tetramers are able to interact when they are triggered by an inflammatory signal and produced by the glial cells, are able to interact with the protofibrils of A beta and increase the deposition of the A beta plaques, and this in turn increase the inflammatory signal that in turn increase the production of S100A9, and then we enter in a vicious circle. Okay, but S100A9 is also a well-known player in inflammation. Actually, complexes of S100A9 and S100A8 are expressed and released at inflammatory sites. So there is a strong correlation between these two molecules and inflammatory disorders. And in fact, in multiple sclerosis, the Q compounds, quinoline 3-carboxamide compounds, which are targeting S100A9, are being explored pharmacologically for multiple sclerosis. You might have heard about laquinimod from Teva, for instance. This is a molecule which is a S100A9 inhibitor. So, okay, we were wanting, we really wanted to have an independent confirmation of this interaction between ORI 2001 treatment and reduction of S100A9. So for that, we were deciding to go for an experimental autoimmune encephalitis mice model. In this model, which is a well-known and recognized model for multiple sclerosis, S100A9 has been described to be upregulated. Okay, this is a very classical model. You basically inject the animal with a peptide, which is triggering an autoimmune encephalitis. So you insult the animal, and then you wait till the onset of the symptoms, and then you treat the animal for 2 weeks, and you see what happens. There is a well-known clinical scoring system that ranges from the death of the animal to different degrees of paralysis, and you can score the animal, you can score the potential rescue of your drug. So when we treat those animals for 2 weeks with our compound, what we get is basically a very profound protective effect. We see that the animals treated with our compound at 2 different degrees or at 2 different doses are really showing a reduced disease incidence and severity. We hardly see some loss on tail tonicity. So this is also confirming the relation between our treatments and the capability to influence on S100A9 driven phenotypes, and it's also opening the door for LSD1 inhibition to be a possible treatment in multiple sclerosis. So where are we standing now on this with this drug? As I said, we have been working very hard in the last years to develop molecules which are basically having all the features that you need to become real human drugs. So right now, ORI 2001 is in the middle of a phase 1 study in an hospital in Barcelona, which is a double-blind, parallel, ascending, single, and multiple doses trial. So the part of the single ascending dose have already finished. We are halfway of the multiple ascending dose. So far, everything is going very, very nicely. So if everything goes smooth, we should finish this multiple ascending dose by December, and by the end of the next first quarter to next year, or the second quarter next year, we should be able to have the whole package ready for starting a phase 2, not only in Alzheimer, but also in multiple sclerosis. Okay, so with that, I am going to make a mention. This is a work that was starting in 2005, 2006, in a seminal collaboration with Isidro Ferrer, where we were, and in those years, we were identifying that UCHL1 was decreased in some areas of the brain with some specific pathologies. UCHL1 was leading us to LSD1 later on. So from these seminal observations, we have been also working with several groups. I am especially grateful to the group of Mercè Pallàs at the University of Barcelona, where we have been developing the SAMPIT model, but we have been also working in lately also in the multiple sclerosis with some groups, some people from the CSIC of Granada, also from some people in the Autónoma and in the UK, and also all the people that you see here from Oryzon Genomics. So thank you very much for your attention.